Hello and welcome to today's podcast. I'm Jimena Ibagon, a PhD student at the Stein Monogastric Nutrition Laboratory at the University of Illinois. And I'm thrilled to have you with me as I share some findings from one of our experiments. Today, we will be discussing how reducing the particle size of field peas increases net energy and improves starch digestibility when fed to growing pigs. So now let's start with a brief introduction to field peas. Field peas are pulses from the legume family and are considered as a cool season crop. This means they particularly grow well in colder climates and are resistant to certain diseases. What makes field peas unique is their ability to thrive in environments that are often too cold or hostile for crops like soybeans or even corn. Field peas are mainly cultivated for human consumption, and in the last 2 decades, countries like Canada, Russia, China, and the United States have emerged as the largest producers of field peas, together accounting about 60% of global crop production. Now, while the nutritional value of field peas for swine diets has been previously reported, it is important to recognize that the quality of these field peas can vary significantly. Factors such as climate, cultivation methods, the specific region where they are grown, and breeding varieties all play a role in this variability. One often overlooked factor that can change the nutritional value of an ingredient is the particle size, the size of the feed particle after grinding. Grinding ingredients is a common practice in animal nutrition. It allows us to create an uniform feed mix for animals. And one of the major advantages of grinding is that improves nutrient digestibility. This happens because the process increases the surface area for digestive enzymes to interact with the feed, breaking down the nutrients more efficiently. However, this process can also lead to higher feed cost. Indeed, data have shown that reducing the particle size of ingredients like corn and lupins can boost the digestibility of both starch and energy. But here is where our study comes in. There is very limited information about the impact of reducing the particle size of field peas, particularly in relation to starch and amino acid digestibility in group house pigs. Also, there has been much comparison between peas grown in the United States and those grown in Canada. So the objective of our experiment was to test the hypothesis whether the particle size and growing location affect the apparent ileal digestibility of starch, amino acids, and energy concentrations. For both experiments, we used field peas from the US and from Canada, and in total we had 5 batches of field peas, one from the United States ground to 600, 400, and 200 microns, and 2 batches from Canada ground to 400 microns approximately. In the first experiment, we formulate 7 different diets. This includes a basal diet made of corn and soybean meal, 5 diets containing 50% field peas, corn, and soybean meal, and a nitrogen-free diet for calculating endogenous losses. For the second experiment, we used the same diets with the exception of the nitrogen-free diet. For the first experiment, we used the Latin square design with 7 diets and 7 periods. We worked with 7 cannulated pigs, each weighing around 60 kilograms, and collected the samples over a 7-day period, 5 days of adaptation and 2 days of ileal digesta collection. The second experiment took place at the swine calorimeter unit at the University of Illinois. This unit is equipped with 6 calorimetry chambers, each designed with an upper part for the animals and a secondary section, which is the bottom part of the chamber, to collect feces and urine separately. For this second experiment, we used chamber as the experimental unit, 24 growing pigs that were housed 4 pigs per chamber with an average initial body weight of 30 kilograms approximately, and the experimental design was a Latin square with 6 periods and 6 chambers per period. Pigs were fed the experimental diets for 13 days, with the initial 7 days being the adaptation period to the diet. The 6 following days were for collection of feces and urine, and we record the oxygen consumption and the production of methane, carbon dioxide, and on day 14, pigs were deprived of feed for 36 hours, and we collect the data for the gases and collect the urine, and this data was used for the calculation of heat production. Now let's move on to the results of the apparent ileal digestibility of starch and amino acids. And first, I would like to talk about the apparent ileal digestibility of starch. Here, We can observe a linear increase in the particle size that was reduced when field pieces were ground from 600 microns to 200 microns for the United States source. And on the other hand, the field piece from the United States ground to 400 microns do not have any difference compared with any of the field piece grown in Canada. Now let's move on the digestibility of amino acids, and today we will be focusing specifically on lysine. Our data showed no significant effect on particle size or growing region on the digestibility of lysine. In fact, we didn't observe any notable difference in the digestibility of any of the indispensable amino acids related to particle size or the region where the peas were grown. And this data aligns with results where particle size was reduced in corn and there was no effect on the digestibility of amino acids. So we came with some conclusions from the first study, and the first one suggests that while reducing of particle size of field peas can improve starch digestibility and therefore energy concentration, the growing location may not have any impact on it, and that the reduction of particle size of field peas may not have a significant impact of amino acid digestibility, which has been demonstrated in corn, as I mentioned. Now let's talk about the results for the net energy in diets containing field peas, and here you can observe that the net energy values didn't change across the diets when we use peas from the United States or Canada. However, we observe a linear increase in the net energy when the particle size of field peas was reduced from 600 microns to 200 microns. This is a key finding because when the particle size was reduced from 400 to 200 microns, the net energy values were similar. This suggests that grinding peas to 200 microns may be not necessary to achieve maximum net energy and potentially will save in cost. Another important observation is that the net energy value of the basal diet did not differ from diets containing field peas, and this means that including up to 50% field peas in corn and soybean meal-based diets does not significantly impact the net energy of our diets. Let's wrap up by revising the net energy value of our ingredient, the field peas. We observe a tendency of greater net energy values in field peas in the U.S. source and a clear linear increase in the net energy when particle size was reduced from 600 microns to 400 or 200 microns, which is a similar story than when field peas was added to the corn soybean meal diets. To summarize, our key conclusions were: first, that reducing the particle size of field peas enhances starch digestibility, leading to increased energy availability, much like what's seen with other legumes and cereal grains. Second, the amino acid digestibility of field peas was not affected by the particle size reduction or by where they were grown, which aligns with similar results in corn, where particle size did not impact amino acid digestibility. Third, reducing the particle size to 400 microns is an efficient strategy to improve energy digestibility without the additional cost of grinding to 200 microns. And lastly, whether ground to 400 or 200 microns, field peas can be added to swine diets containing corn and soybean meal without compromising the energy value of the diet. With that, I would like to thank Swine Innovation Pork for the funding and the Stein Monogastric Nutrition Laboratory for the help in conducting this experiment. Thank you for joining me today, and I hope the discussion provides some valuable insights into the effect of particle size and growing region of field peas. If you want to know more about our research, you can go to nutrition.ansi.illinois.edu. Thank you.